logo eu começo a falar diferente. Começando a gravação em 3, 2, 1. Good morning, everyone. It's a pleasure to present today's lecture. We'll have an international guest here. We will talk about prenatal stress and neurodevelopment today. What do we know? So we'll have as a speaker Professor Sebastián Illanes López. He is Professor of Medicine at the University of Los Andes and Research Director of Obstetrics and Gynecology at Clínica Dávila, Santiago. He's adjunct professor at Wayne State University, USA, and holder of the S. Shan Ratnam Professorship at the National University of Singapore. He's founder of the Perinatal Medicine Unit at the University of Los Andes, and his research focuses on placental-related diseases, developing biomarkers to predict pregnancy complications and preventive strategies. He's a former Vice Dean of Research at the University of Los Andes, and he has led major national and international societies in obstetrics, gynecology, and prenatal diagnosis. After the lecture, we'll have some comments by Professor Helena Paula Brentani. He's a psychiatrist. She has a master's in psychiatry and medical psychology from FMUSP. She has a PhD in psychiatry from FMUSP, the University of São Paulo. She's an associate professor at the Department of Psychiatry here at the University of São Paulo. She's an advisor for the Interunit's graduate program in bioinformatics at the University of São Paulo. She's also the coordinator of the Autism Spectrum Disorders Program, PROTEA, and of the Biobank of the Institute of Psychiatry here at the Hospital das Clínicas, FMUSP. She's coordinator of the Systems Biology and Psychiatry Laboratory, SAISBio, and she's an associate researcher at the Brazilian Center for Applied Research in Early Childhood. So, Professor, it's an honor to have you here. I would like to welcome you in the name of our department. Let's begin the presentation. There you go.
Okay. Well, thank you very much for your very kind introduction. I'm very, very happy to be here. This is my second time in Sao Paulo. And I'm very happy about the potential collaboration that we can build with the ongoing collaboration, but also to increase the amount of knowledge that we can build together. So let's start. All this audience know that there is a huge evidence, a growing evidence, indicating that maternal stress increase the risk of having adverse neurodevelopment outcome in the offspring. The amount of papers that have been published, the first one was in 93, until now is huge. And only this year, we have more than 56 papers relating only with prenatal stress and neurodevelopment. And this started with fetal programming. I don't know if you are aware of the story of fetal programming, and this is the story, the Market Garden operation. This was an airborne operation, the biggest one in the Second World War, were led by the British, and the idea was to take all the bridge between France and Germany, and they had to take all the bridge in order that the tanks advance quickly to surpass the Rhine and reach Germany. And what happened is that they have some success in the first bridge, but they couldn't cross the last bridge in Arnhem, and then it was a failure, a complete failure. And what happened is that the German retreat to Germany and left some cities in Netherlands without any food, okay? Because it was a very, very, very rough winter. And that means that there was a huge hunger in Netherlands at that time. And this was a very special type of hunger, because it was a hunger that only lasts 3 months, the 3 months of the winter, because this was the time that the Allies were able to bring food to these people. And what happened with this cohort of babies that were born, or were they were in the womb of the mother during that time, that then Professor Baker took this cohort 50 years later and developed the Baker hypothesis, where he said that intrauterine nutritional environment is determinant for the risk of developing metabolic cardiovascular disease. Okay, and this is the prenatal exposure. That is, in this case, it will be at the beginning, it was related with maternal body mass index, placental microgranules, fetal growth restriction, produce a pre and prenatal molecular processes that produce abnormal postnatal outcome. And what happened in the uterus is a disruption of the epigenetic profile that produce a predisposition to adult disease. The amount of literature showing that condition linked between prenatal stress and depression is huge. I wrote some of them, showing lower Brazelton RNA test score in the children exposed to prenatal stress, sleep problem in infant, higher anxiety in infant, more difficulties in temperament, lower cognitive ability, etc., etc. But it's interesting that after this initial literature, more literature comes that is not the same if the baby is a boy or is a girl. And it seems that the placenta, being XX or XY, produce a modification between the messages that are brought from the mother to the fetus. And this is not the same. And the type of neurodevelopment disorder that the boys and the girls develop, depending on the stress, is different. So, in 2019, the CDC had a webinar calling Protecting Pregnant Women and Babies During Public Health Emergencies, because that is something that produces stress in the mother. And at the same time, we were publishing this paper, and it was the first paper that we published related with this topic, when we wanted to see the effect of the earthquake in perinatal outcomes. And at that time, I was working in a big hospital. We had 8,000 deliveries a year. Well, it's a big hospital for Chilean standard, probably here in Sao Paulo are bigger than that. But what we did is that we took all the babies that were born in 2009 and 2010 in that hospital. So we did a case control study, and we compared those that were born in 2009 with those that were born in 2010, and we divided these babies if they were exposed to the earthquake that was in February 2010 to in the first, second, and third trimester of pregnancy. And what we discovered is that the size of the baby, if they were exposed in the second and third trimester, impacted males and females, and they were shorter if they were exposed to the earthquake, but also the head circumference if they were exposed during the first and third trimester, but only males. That is interesting in terms of what we will see later, that this dysmorphism between sex and the head circumference is important for the data that I will show later. And also, the gestational age at delivery also was reduced in the babies that were exposed to the earthquakes. So we decided then to apply to a test that was developed by the educational school in my university. And this is a platform, a web platform, that evaluate and assess the early readings and math skills of the children when they enter the school. So we did a pilot study looking at children that were born or were in the womb at the time of the earthquake. And when we applied the DIA, the non-exposed have a much higher score. So we did a study that we published a couple of years ago. So we took this test that is done to many children in many schools in Chile between 2015 and 2017. And we tested more than 3,000 children. And of those, 2,400 were non-exposed. There was those that were entering the school 2015 and 2017, and almost 900 that were entering the school in 2016, 6 years later after the earthquake, so that they were in the womb of their mother during the earthquake were used as the cases, and the other ones are the control. And what we found is that the children that were in the room that were exposed to the earthquake have almost a 30% lower in listening comprehension, 30% lower in letter knowledge, and 12% lower in beginning sound recognition. So there was a huge impact. 30% is a huge impact. It's the same difference that we have from very poor areas of Santiago, the schools, with a very high society schools in other parts of Santiago. 30% is a very, very big difference. And here I only brought some of the graphics of this paper showing that the listening comprehension in the non-exposed versus the exposed, and there is difference also depending on the trimester that where the children were exposed, and also there is difference between males and females. It's not the same, the different skills in math and in reading, depending if it's a male or a female, and depending on if it was exposed in the first and second trimester. I don't want to go into details, but there is difference between the different skills that you are testing, and there's difference between the trimester where the children were exposed. And at the same time, this paper, I think, is a breakthrough paper, because this is the first paper, or one of the first papers, showing that this is not only a correlation between maternal stress and psychological or cognitive changes in the children. This is a paper where they were showing that the MRIs of these babies in the womb, in mothers diagnosed with anxiety and psychological distress, already have anatomical changes. So when there is an anatomical change, that means much more interaction between what is happening in the womb, because it's producing an anatomical change. And the changes that they were able to demonstrate was that the anxiety was associated with a smaller fetal left hippocampal volume, and the maternal anxiety and stress were associated with increased fetal cortical gyrification in the frontal lobe and temporal lobe. So there is a change in the structure of the fetal brain because of this psychological distress of the mother. So in terms of fetal programming, we know that neurodevelopment disorder depend, and there's a lot of research looking at the association with obesity, hypertension, type 2 diabetes, but now we know that these neurodevelopment disorders that have at least some evidence of anatomical changes also are related with prenatal distress. So what is the size of the problem? The size of the problem is that, at least in US, 1 in 5 women will experience some sort of psychological distress during pregnancy or postpartum. And in Chile, this data of Chile is before the pandemic. The number are worse now. In 1 in 3 will experience some sort, and this is a paper published some time ago, but showing a very high level of distress or psychological distress during pregnancy. So the big question, and this is one of the line of research that we have, is how this maternal stress is transferred to the fetus. Because if there is an association, we need to understand how the maternal brain is transmitting this stress to the fetus. And there is a lot of mediators. And there is a cytokine, tryptophan, cortisol, ROS, catecholamine, microbiota, and they are poorly understood, and the research doesn't show that they can account for all the changes that you observe in the fetal brain. So we proposed as a hypothesis that perhaps one of the molecules that we were studying, that are the extracellular vesicles, could be cell-to-cell messengers and could be the responsible for the communication between the maternal brain and the fetal brain. And we tried to demonstrate that these small extracellular vesicles were a way of brain-to-brain communication. And we will try to demonstrate you with the next slides that this could be the case. So these extracellular vesicles allows the transfer, they are cargo, they are vesicles that are cargo for protein, lipids, RNA. They are complex packages and can contact distant cells. So we took advantage of the research of a neuroscientist, Ursula, that published in 2018 a very interesting paper that in rat serum, there is astrocyte-derived protein biomarkers when you stress these rats. So the conclusion of this paper is that neural cells, astrocytes, release extracellular vesicles into the peripheral blood. And that these vesicles, if the animal is stressed or not stressed, are different, have a different cargo. So we took that hypothesis and we said, okay, this could be a way that this brain of the mother is communicating the brain of the fetus, the stress, and producing neural anatomical changes in the brain of the fetus. So our hypothesis is that the astrocytes of the brain of the mother release circulating extracellular vesicles that go to the plasma and reach the placenta, change the function of the placenta, and reach the amniotic fluid and reach the brain of the fetus. This is where our hypothesis. So we develop a model of mild stress. There's a mild prenatal stress. This is a model that is very well known. And what you do is in rats, you take the pregnant rat, and between day 6.5 and 15.5, you do a handling and restraint for 2 hours a day for 9 days. And then you evaluate the stress-induced anhedonia. So the idea is that the rat that is stressed doesn't care if they take water or water with sucrose. Usually the rat, when they're not stressed, prefer the water with sucrose. And this is a sign of anhedonia, and this is a sign of mild prenatal stress, that it is the restraint paradigm. And what we found is that the fetuses of the stress group were smaller, the fetal length was smaller, the placenta was smaller too, and the adrenal glands were bigger, and the blood vessels in the adrenal gland were bigger, and the cortical and medulla of the stress were absolutely different between the maternal adrenal glands. And we also studied the EVs of these animals. And what we found is that the stress animal in the plasma of the mother, there was a huge increase in the amount, on the total amount of extracellular vesicles, but also these extracellular vesicles had a very different cargo. So, meaning that the stress that we were producing was changing the amount of extracellular vesicles, but also the cargo of those extracellular vesicles. And we did a biodistribution of these EVs. So we took donors, controls that were pregnant rats, not stressed, and the stressed one. We took the plasma extracellular vesicles, and we put a deer that is a marker to be able to track those extracellular vesicles. And then we injected the extracellular vesicle from the controls and the extracellular vesicles from the stress into controls and into stress. So we had 4 groups: the control with control extracellular vesicles, control with stress extracellular vesicle, stress with control, and stress. Okay? And we look at the placenta and the fetus, and look at what happened. When you have a stress animal with a stress extracellular vesicles, the amount of vesicles that are captured by the placenta is increased. But if you look at the fetus, the stress condition produce a huge pass of extracellular vesicles that are reaching the fetus. Okay, so it seems that the placenta is capturing a big amount of the increased amount of extracellular vesicles that are being released by the brain of the mother, of the rat, But anyway, a huge amount of these extracellular vesicles are reaching the fetus. So what we have done later, and this data is not published, we took the rat and we did an experiment is called electroporation. So in a pregnant rat, we do a surgery, we take the uterus and we do with the two electropores, we do a magnetic camp and we inject a deer. So this deer is included in the astrocyte of this pup, okay? And then we take this pup, we allow them to birth and then to growth, and the female ones that have the deer in their astrocyte, they're adults that are releasing extracellular vesicles that are marked with a deer. So we are able to track the extracellular vesicles released by the astrocyte, and then we made them, and so we are able to look at the pups or the fetuses inside the womb in a mother that is able to release extracellular vesicles coming from the astrocytes. Okay, so this is what is happening. The mother, the female has the astrocyte with the extracellular vesicles that are marked with the deer, that are released to the circulation, and they are reaching the fetuses. And what happens is that you start to see these extracellular vesicles that are in the amniotic fluid, but most importantly, they are in the fetal brain. So what is happening is that we were able to track extracellular vesicles coming from the astrocyte of the mother going to the brain of the fetus. And we think that because of the cargo of these extracellular vesicles, we have the transcriptomes of these extracellular vesicles change. We think that this is at least one of the mechanisms in how the mother is able to transmit the neuroanatomical changes in the fetus. So what we have done later, we have done some in vitro studies, taking astrocyte, we culture them to obtain the extracellular vesicles from these astrocytes, and we do culture with corticosterone levels or not, trying to mimicking a stress condition. And then we take the neural stem cells from the brain of the fetuses and we do co-cultures of these neural stem cells monolayer culture with the small extracellular vesicles coming from corticosterone or not corticosterone cultures. And what happened is that the proliferation is reduced, so you have less neural stem cells in the culture, but most importantly, there is an increase in maturation. So what is happening is we believe, and this is our hypothesis, and this is the ongoing work, we have a grant, we just won a grant to look at this, is that in a condition of stress, what the mother is saying to the brain of the fetus, you have to mature earlier because you will be born earlier and you will face an adverse environment. But what happened with early maturation, you reduce the capacity of this brain because there is a early maturation and you don't allow the full maturation of the brain in the times that are normal. So, and we think that this is potentially the explanation of why you see cognitive and abnormal cognitive development in those children, because they have a mature or more mature and less proliferative brain. So this is an ongoing work. This work is being done in the lab of, we are working, this is a collaboration with the Neuro Lab, and this is that is directed by Federico Batis and Mario. So our restraint stress protocol did not reduce placental weight, but induced fetal intrauterine growth restriction, meaning that there's not an hypoxia fetal growth restriction. It's a placenta that is not working properly due to the extracellular vesicles coming from the brain of the mother, that maternal astrocyte-derived rich placenta and fetal brain, and suggests that astrocyte-derived stromal extracellular vesicles can modify the biology of neural stem cells. So what we are doing now, what are our ongoing plans? So we have a cohort of patients where we have the patient, We are evaluating them mainly at 32, 36 weeks. Our idea now is to expand this also to 24, 28 weeks, where we are doing maternal tests for depression, stress, and anxiety with different tests. We evaluate the patient at delivery, and we take cord blood, and we are following the children at 6 months, 12 months, and the idea is to follow the cohort until 5 years. And we are doing evaluation of cognition, language, motor, social, emotional, and adaptive behavior with different tests. And at the moment, we are in this process, and we have already recruited more than 1,000 patients. So we have the data of this, and we have the samples of those patients. We have the maternal sample, and we have the cord sample. We have the ultrasound of these babies, and we have the test done at 3 months and at 6 months of age. So I will show you some of the data that we have that are very interesting about the how. Because if we want to know what is happening in the brain, we cannot do a biopsy in the brain of children. Our ethical committee doesn't allow us to do that. So what we have to do is to use the cord blood, because this is a sample that we can take, and we can use this sample to evaluate the brain of the fetus, because we can pull down the extracellular vesicles that are coming from the brain of the fetus. So we have mother that, because of this psychological distress, perceived stress, anxiety, or depression, depending on the test that you have done in her, you can qualify her in high or low maternal psychological distress. And we are looking at the cord blood. We are isolating small extracellular vesicles, and we are evaluating these children with the different tests at 3, 6 months, and our idea is to follow them until 5 or 6 years of age. And we are evaluating neurodevelopment trajectory and temperament, and we are using proteins and microRNAs in the small extracellular vesicles to try, using the cord blood, to predict what will happen with these children. What is the idea? If you are able at delivery, because what's happened is that we will not be able to change the anxiety or the distress of the patient. That is very difficult, because in a city like Santiago, how you isolate the patient from the city, from the family, from all the stressor that she has surrounding her. But there is opportunity that the brain of the fetus is not fully developed until 2 years after birth. So if we have a biomarker in the cord blood that is predicting the temperament or the neurodevelopment trajectory that this children will have, we have an opportunity to tell the parents or to tell the health system, oh, look, this baby will have a 30% less listening comprehension. We have to intervene now. So what we have found is that at 3 months, we are evaluating urgency, effortful control, and negative affectiveness. These are the things that you can evaluate. And what we have found is that the children that were coming, or that are coming from mother with a high prenatal distress, have more negative affectivity. That is very statistically significant. And this is prenatal stress and depression. And at 6 months of age, not only negative affectivity is positive, but also urgency. Okay? So this is an ongoing research. We have not published this data. This is a very new data that I'm showing you. I think you are the first or second one audience that is looking at this data, but we are very happy that our model at least is showing, and we are able to detect this difference. And what we have done is we have looked at the concentration and the size of these extracellular vesicles, that there is no change in the concentration and in the size, but when you do the RNA sequencing of these extracellular vesicles, we have found that there is some microRNAs that are negatively correlated with the negative affectivity at 3 months of age and also at 6 months of age, but only in boys. not in girls. And that, if you remember the first slide showing that the boys were more affected, we think that these microRNAs are showing us something. We need to validate this result. We need bigger cohorts to see if this is the case, but we are very enthusiastic that this could be a path, that to evaluate the children at the time of delivery in the cord blood to predict what will happen with them in terms of neurodevelopment trajectory and neurodevelopment as a whole. In this study, we also found that one of the microRNAs that we were more interested on is the microRNA 2003, because these have shown that in other study that is related with neurodevelopment. And we test in our cohort, and women with the Edinburgh score over 17 showed increased levels of this microRNA in the cord blood. Interestingly, we then went to see the mothers at the time of breastfeeding at 3 and 6 months. And the mother that have pain, mastitis, and have a very difficult breastfeeding also show an increased levels of microRNAs in the cord blood. We don't know what that means, but it's an interesting finding. And we have started to look more in deep in what is happening in the breastfeeding. So we have started to look at the milk as a source of extracellular vesicles to see if we can understand better two things. One, a predictor of what is happening in the mother in terms of psychological distress, but also how the mother that is stressed is releasing extracellular vesicles in the maternal milk, and this maternal milk is reaching the baby, and what is happening in the newborn with these extracellular vesicles that are coming from a brain of the mother that is already stressed, so they have a different concentration and also they have a different cargo. Because we know that these milk extracellular vesicles that are released from the reach all the gastrointestinal tract, and there is several publications showing that the extracellular vesicles and their content, especially microRNAs, can reach the intestine, can modulate the immune system, can regulate metabolism, and can reach the nervous system. So what happens if a mother is breastfeeding, and in her breastfeeding you have extracellular vesicles that have a different cargo because of her stress. What is happening in the newborn? And this is what we have been studying. So this is a small cohort also. We have 19 control and depression, and the only thing that is different is the Edinburgh test. One have a score of 5 and the other one 14. And we took milk samples at the first time in the morning and processed it the same day. And in this mother, no medication at all. So there were any mental health medication. And we did RNA sequencing, is the more extracellular vesicles, and we found that there is a huge amount of genes that are upregulated and downregulated in the extracellular vesicles coming from a mother that have prenatal distress. And interestingly, most of them are related. When you do a gene ontology analysis of this type of genes that are upregulated and downregulated, many of them are related with neuron development differentiation. Okay, so that is interesting, because if these extracellular vesicles that are in the brain, that are reaching the intestine of the newborn, are reaching the brain, we have some of the genes that are upregulated or downregulated that are involved in neuron development differentiation. So again, we have found in the extracellular vesicles this biomarker, and we want to see if in the milk this is also a biomarker of the behavior of the children later on. And this is the new project of Reina. That is the one that Reina, she's the one that has been doing this research in my lab. She's my postdoc. And now her new research is to look in an animal model, what happens if you have a foster mother and a foster dam, and we will take the mouse milk, and we will see what happens if you give this milk to a non-stressed mother, or you give, so we will do all the analysis in the brain and the structure and neuroinflammation and biodistribution. So the idea is to see if our hypothesis that we have shown in our previous data can be demonstrated in an animal model. So, and this is the last slides. All of you know that there is a huge amount of neuroanatomical changes that are observed over the course of the human pregnancy. And this is a study that appeared, I don't know if you are aware of this study, it's a very nice study, published in August last year, so 1 year ago. And this is a neuroscientist that she wanted to become pregnant, and she did a study of N of 1. She was the only patient in that study. And she had 25 MRIs, 5 MRIs before pregnancy, 15 during pregnancy, and 5 post-pregnancy. And she measured all the levels of sex steroid hormones during this period. And she had, this is the data, and showing the individual tracks and the changes in the different, this is the the first, the second, third trimester. So the 3 in the middle are the MRIs done during pregnancy, and these are the postpartum. And this is the most striking graph, showing the gray matter volume and the total brain volume. And I want to focus in these two, showing that the pre-pregnancy gray matter volume and the total brain volume was higher, is reduced during pregnancy, reaching the lower level at birth, and after 2 years without recovering to the previous stage. So this is a huge change, huge change. There is more and more data showing this data. So this is interesting, but it's very difficult to correlate this with pathology because we need to do MRIs. So we thought in our lab how we can overcome this problem. And we designed a study where we took spinal fluid of the patient. So the patient arrived in labor or with a elective cesarean section, and when the anesthetist did the spinal anesthesia, we asked them to give us 5 ml of spinal fluid. So we have spinal fluid of women in labor and not in labor. And what happens is when you do the RNA sequencing is absolutely different. The gestational age is not different, but the RNA sequencing of the brain of a mother in labor is absolutely different from the brain of a mother not in labor. And we have more than 240 genes that are downregulated and 95 upregulated when they are in labor. We are interested specifically in long non-coding RNAs. And interestingly, what happened is that those genes, when you do the gene ontology and you look at those that are enriched, you have neuronal ion channel clustering and neural stem cell division. But when you look at those that are downregulated, you have neuroapoptotic processes and neuron death, showing that probably we have in the RNA sequencing of the spinal fluid a reflection of what is happening in this reduction of the gray matter. We believe that is our hypothesis. So what we are doing now, because we cannot do this because we don't have money for doing MRIs, but we have money to do blood. And what we are trying now is to look at the long non-coding RNAs or the RNAs that are in the spinal fluid, if we can find them in the maternal blood. Because if we are able to find them in the maternal blood, we can measure them in the first, second, and third trimester and try to mimic what you are seeing in MRI with a biomarker, a cell-free RNA biomarker in the maternal blood. So we are trying to develop a non-invasive well, it's invasive because you have to take blood, but a blood test to evaluate the function of the fetal brain. And we want to do this in a normal cohort first, and then in a cohort with depression, with prenatal distress, with postpartum distress. So this is the work that we are doing now, and we are applying for funds for doing this. So this is my team. And these are the collaborators. Well, Reina was a collaborator, but now she has just been contracted by university. She's working in my lab now. And that's it. So I'm very happy to answer questions if there is any. You want to go there? Okay.
Pode sentar aqui. Just let me change the camera. Okay. So thank you, Professor, for your brilliant lecture. It was wonderful to hear about all these studies going on, quite interesting. Now, Professor Elena, if you could please share with us some thoughts of yours.
Good, thank you. And first of all, thank you for your speaks. It was very good and very, very interesting. And we have been doing a similar work with a small cohort, as you already know. And there are things that I think I would like to ask about your thoughts. First thing is in 3 different studies, we saw differences between depression and anxiety. And so we thought about psychological distress, but as I've been studying, I think that they are completely different and not the same thing. And even the biological markers will be different. And so I would like to know what you think about, because you saw in some studies you talk about depression and others about anxiety. And
I absolutely agree with you. Probably what happens is we put them all together because the size of our doesn't have enough numbers at the moment. The new cohort that we have is bigger, but the data that I showed is from a smaller cohort, and we were unable to divide them between depression, anxiety, psychological distress, because we didn't have the numbers. Also, you have to take into account that when we are talking about Edinburgh test, the sensibility of the Edinburgh test is very low. It's not good. So this patient doesn't have a psychiatrist evaluation that tell me for sure that we are only looking at psychological tests that doesn't make a true diagnosis. They are screening tests. So I think most of the variability that we have are because the diagnosis of the mental health condition of the patient is not very accurate. And I think one of the ways to improve this is to do better evaluation of the mental health situation of the patient in order to understand better the biomarkers that you are having in the maternal blood or so yeah, I think this is a necessary step. But on the other hand, it's very encouraging that even though we are putting clinical conditions that are very different, we still see an impact that can be. So I will try to highlight more than the specific RNA that we are looking at, the concept that it's possible to look at the core as a source of biomarker to predict what will happen with the children. So what we need to do is bigger studies where we evaluate better the mother in terms of mental health during pregnancy, we have the biomarkers, and then we follow up the children. Also, the when I spoke with the infant psychologists that were doing the studies at 3 months of age, said, "Oh, how you can tell me that these children have a different temperament of this one?" They were filming them, and for 1 hour how they play with the mother, etc. And they reach conclusion with that, but I think the evidence is not very solid. As I'm a gynecologist and I'm a researcher that I want more strong data. So what I think we need to do is to wait more to see what is happening with these children when we have tests that are more robust to evaluate the psychological behavior of the children later on in life.
Sure. And I agree with you, totally agree with you. But I would like to know how you think and how you are doing these things. And other thing that is very interesting that we have been performing is we look to cortisol, we look to cytokines during pregnancy and their interaction. And then we evaluated the methylation of HPA genes. And I was at the head time very interested in oxytocin. And then we published 2 papers showing that we can program the oxytocin gene methylation in the baby with 12 months. And so I'm very interested also in understand not all the HPA and cytokines programmation, but also the oxytocin programmation as a modifier of how I see the world, not linking to the motherhood and nothing like that, you know? And like how can I predict the world is there? And so at the end, we have been studying this a lot here, and I will show you more. But and have you thought about these or looking about?
You have to remember that I'm not a psychiatrist, I'm not a neuroscientist, I'm a gynecologist. And I become interested in this because of biomarkers. My main career have been in biomarker of preeclampsia, biomarker of fetal. Now I'm working with Andrea in biomarkers of hypercholesterolemia in pregnancy. And I found that it's amazing that we are in the 21st century and we still don't have biomarker and proper biomarker for psychiatric disease.
Do you know what are psychiatric diseases? I have this question.
Or mental health disorders.
Yes, sure.
Okay, so I think we need biomarkers and we need biomarkers also to predict mental health disorders, because if I take all the prevalence of diseases in pregnancy properly, nowadays mental health disorders are the most important ones in pregnancy. 1/3 of my patients, at least, are using Actulin. 1/3. It's much more higher than preeclampsia, gestational diabetes. So we need, and there is no enough psychiatrists to see all the patients, because you already have a lot of work with patients that are not pregnant. So how we help you? And my idea is that the psychological test, when I saw this was happened 4 or 5 years ago, I was studying the sensitivity and specificity of Edinburgh test, because they want to apply to all patients in Chile. Say, okay, let's see what they do. And then it's awful. The sensitivity of Edinburgh test is very bad, very bad. So I thought, well, can we do better? We cannot have a biomarker that predicts better than the Edinburgh test mental health disorders. And then we started to do it. And then we had the COVID pandemic, and was the perfect time to do these studies, because the increase of mental health disorders, at least in Chile, imagine that here in Brazil was the same, was huge. So we had a COVID cohort, and then many of the data that we have from the cohort and the data that I showed was from this COVID cohort. We had around 200 patients. And then, well, that's it. But But my idea, my focus is to have biomarkers in pregnancy for mental health, or that predict mental health disorders in the mother and in the fetus. This is my main goal.
Okay. Great. Well, I would like to collaborate with you. You will have some samples
Absolutely.
here, and
I'm
we'll
very happy
discuss
to do
about this tomorrow. And I don't know if do you have any question, any comments?
Thank you very much for being here. I appreciate it. I just have to leave for a meeting. So
Okay.
appreciate a lot your visit. We are honored to have you here, your team. Considering that some of this is a very interesting research, very heuristic findings. Considering that some of them will be replicated, and so suddenly you have a biomarker or that you predict early neurodevelopmental problems or even mental disorders in the future. How you consider early interventions? What kind of early intervention we made for someone who has a positive biomarker?
We have done a small study. We haven't. I didn't show the because I saw that my presentation will be long, but it was short. Okay, but I didn't show the data. But in the same study where we looked at the DIA test, the test to evaluate the reading and mathematical skills, we have the same cohort after the intervention that were done because of the poor performance in the test. Okay? And the children that were scoring lower because they were exposed after the intervention, they reach the same level of the children that were not exposed. So we closed the gap. It wasn't absolutely close, but we reduced a lot the gap. It wasn't a 30% difference, it was 10%, 5%. So what we believe is that probably we don't need to do a huge intervention. It's only very focused intervention in reading skills, because there is other problem that the mother that have a stress, the stress continue to be after delivery, during breastfeeding, and during all this period that the children need more input from the mother, from his mother. So the intervention could be at two levels. One, to intervene the mother, to try to be able, if we have better tests, to recognize the patient that have a mental health disorder and to improve and treat those patients. And also those patients, babies that have been exposed to a mother with a mental health disorder, to try to do specific things like to improve the face-to-face communication, to improve the bonding, to improve the at later age, the pre-reading skills. I don't think you need to do very complex intervention because the very simple interventions that we did in our small cohort were very successful.
Have one more question here.
Hi, thank you very much. It was really interesting. I don't know, I think I was, my first question was something like just Professor Euripides asked, but I don't know, I'm a little bit concerned because as you said, the stress will be there because normally like here we have an ambulatory for a pregnant woman that lives with this stress. And it's really hard because we see like a psychosocial stress with low income and violence and things like this that will stay in the population, unfortunately. And I was like, imagine like if we could do this biomarkers like more broadly, what would be the impact? Because like I don't know, like these mothers sometimes they are really under a lot of pressure being alive And I don't know, I was thinking like what we could do with
Yeah.
this information to improve broadly.
I understand your question, and I understand perfectly your concerns, and I may share your concerns. I think this is a very difficult situation. But what happened is that I think it's very unfair to be born and already know that you will not reach your full potential because of the stress of your mother. I think there is very little amount of things that are more unfair than that. So, yeah, we need to take care of that. And the first step is to recognize the problem. And to recognize the problem, you have to diagnose the problem. Because what happens is that we don't have enough resources to intervene the population as a whole. So we have to focus our intervention. But to focus our intervention, we have to focus in those that are more vulnerable. And those that are more vulnerable are those that are coming from mother are more vulnerable. So I think one way is to do what we are trying to do, to diagnose those that have we know to predict that we will have a bad outcome. I don't think a biomarker will be enough. I think we will need to use artificial intelligence in order to see clinical variables of the mother, biomarkers, environment, social variables, in order to build algorithms that predict with a high sensitivity and specificity this vulnerability. And in a population, we cannot burden more the already vulnerable woman saying, okay, I'm sorry, your baby will have a 30% less of reading skill when they reach school. We cannot say that. We say, okay, look, we have a solution for you. And that solution needs to come from the government, because these citizens that will be citizens of the Republic of Brazil or Chile in 20 years, 30 years more, you need them with a very good brain to be something, a citizen that will be a set of for a country. So yeah, it's difficult, but the first step, I think, is to make the diagnosis. We cannot close our eyes, because if we close our eyes, the unfairness of this situation will continue.
Any more questions? Anyone?
Thank you very much for your lecture. It's very interesting. So I am a neuroscientist. I work with Elena, but I'm in vitro neuroscience, so neurobiology. But we also have a cohort. We are following the children for 3 years, but we collect the blood or the saliva, actually the saliva and other stool, etc., for the children until the 1,000 days of life. But I'm interested if you are collecting more samples from the children to analyze the microRNA in the future, for example, and correlate with the diagnosis or with the processing of evolution, and also the cytokines of the mothers, because I'm interested in infections during the gestation period, or even the production of cytokines during the gestation period as predictors for epigenetic changes.
Yeah, very interesting question. We have applied for a big grant to do that, and we didn't get it. So at the moment we are stuck, and what we are is to push the cohort bigger and bigger until we have the funds to test the children. Yeah. No, we have the cohort, but we don't have the money to take all the samples. The children are now 4 or 5 years of age, so we will apply new grant now to see if we are able to have the money to test this cohort. But this is something that needs to be done. Need to be done to take the stool to analyze the microbiome. We are absolutely sure that the microbiome has something to do with this. the metabolite that is produced by the microbiome change. Or not only the metabolites, you know, I don't know if you are aware, now it has been described that the bacteria release small extracellular vesicles, are called the bacterial small extracellular vesicles. And we have data from another research that these small extracellular vesicles coming from bacteria are able to modulate the immune system. So what we have demonstrated is that babies that have been born by cesarean section and normal vaginal delivery have different microbiota at 2 weeks of age. And when we take this microbiota and we isolate the small extracellular vesicles of this microbiota, there is more tolerance in the babies born from vaginal delivery compared with the cesarean section. Meaning that the bacterial extracellular vesicles coming from the microbiota are different, and probably these have a huge impact in the neurocognitive development of children. So if you are taking stool, something that we have the technique in our lab to isolate the bacterial small extracellular vesicles, and this could be something very interesting to correlate this the cargo of these small extracellular vesicles with the neurodevelopment. This is something that be very, very interesting. But answering your question, we at the moment we have the core, we have the evaluate psychological test at 3 and 6 months of age, and we are waiting for resources to also have more samples in the ongoing.
We got any more questions? Okay.
Thank you. Sebastián, I heard your presentation several times, but I always learn new things, so thank you very much for that. I want to put you in a different situation, probably a more positive situation.
Okay.
As she said, we live in a stressful environment, and probably a mother that she born from a stressful environment, she will have more anxiety. So probably also their child will have a problem. But in this positive view that we are not doomed, probably is part of the evolution. We live in a stress environment. So which is the advantage of all of this? All the extracellular vesicles coming from the stressed mother, reaching the fetal brain, there's some kind of advantage, not only more anxiety or reading skills delayed, should be an advantage.
You are right. We have thought about that because evolution doesn't happen without a reason, okay? And if we have this evolution and this mechanism where the brain of the mother is communicating with the brain of the fetus and is producing change, is because the stress of the mother have to be communicated to the baby, because this baby will face a very abnormal or very dangerous. But I think this is my belief and my hypothesis that the problem is that these mechanisms were developed for thousands of years, and the risk that the mother was facing 3 or 4 or 5,000 years ago are very different from the stress that we have now. So what I think is that we have mechanisms for those stress, but not for the stress that we are facing now. So the adaptive mechanisms will need to take a long time, and we will not see, we don't have that time because we have the problem now. So our body, our brains are adapted to situations that have changed a lot. The stress that we have now in the cities, the traffic. I was stressed with my arrival today at 5:30 in the morning that I will be able to reach the hotel on time to finish some part of the talk. So I was stressed. And the reason for that is that they had advised me that the traffic in Sao Paulo was very bad. I'm wrong? No.
You're right.
Yeah. So this stress wasn't present 5,000 years ago, and the stress of that mother was if she will be eaten by a lion or not, or if she will have food or not, or the snow that was falling will kill the children. So probably we have an over-adaptation because of the risk that we had in the past and the stress that are now stress, but that trigger the mechanism, but you don't need the consequence of the triggering of the mechanism because the work is not something that will put you in death or life situation. This is my guessing, but this we can discuss all this because this is what I think is happening. Somebody could argue against that.
Sorry, do we have any more questions? So I think that wraps it up. Thank you, Professor. In the name of our department, it's a pleasure to have you here. Professor Elena, thank you for sharing with us some comments. It's an honor to have you here. Thank you so much. I guess that's it for today. Thank you so much.
Can I just
Yeah. Yeah.
Yeah.